Not all genes undergo independent assortment, so we can understand linkage and eventually be able to describe how gene linkage occurs. So, in Morgan's experiments, he knew that Mendelian genetics predicted that all traits will assort independently, that the inheritance of one trait will not influence the inheritance of another trait. But if you recall, Morgan's fruit flies only had 4 pairs of chromosomes, and that led him to make a very interesting discovery that Mendel would not have been able to explain. Not all traits is sorted independently. Even though we expect in Mendelian genetics that genes do not influence the inheritance of each other, Morgan found that they did. And what he was able to conclude is that this meant the genes were on the same chromosome. They were physically a part of the same structure, and we call genes like this linked genes. These genes are physically part of the same chromosome, so they are physically attached to each other. So during metaphase, when all of the chromosomes line up in meiosis 1 and 2, they don't independently assort. They go to the same place. They have to, they're physically connected. So Morgan saw that unexpectedly those genes were influencing one another. Now, it wasn't a case where the gene, if you had the gene for, say, a gray body and normal wings on a fruit fly, those would not always necessarily be together. But if they started together, they had a greater than normal chance of ending together as well. But there was still some recombination. Sometimes you would end up with the regular wings being paired up with the black fly body instead of the gray fly body. This is because of crossover, which we can see in the top of our diagram on the right-hand side. Crossover, remember, is when DNA gets exchanged from one chromosome to the other in the tetrad. What this does is this recombines linked genes. It ensures that not just because the two versions of it, those two alleles are on the same chromosome, it makes sure that they're not always on the same chromosome. It gives a little bit more variety within the offspring pool. And the amount of recombination that occurs directly correlates with how far apart the genes are. If you have two genes that are super close to each other, like gene 1 and gene 2 in the diagram, chances of them crossing over are very low. They're right next to each other. Is it possible for crossover to occur right between them? Yes, it is. They can cross over right between each other, but the chances of it happening is unlikely because the location of crossover within the chromosomes are random. However, gene 1 and gene 3, yeah, they're far apart. They could cross over fairly easily, and they probably do. In fact, if you get far enough apart on the same chromosome and the chromosome's big enough, crossover happens so frequently that you can't distinguish it from independent assortment, so it becomes random all over again. So if two genes are very close to each other, they're unlikely to cross over. If they're very far apart, they are likely to cross over. And we can actually describe this crossover trait. In this example, we're breeding a generation of flies that is 2,300 offspring strong. We're going to take one fly that is a heterozygote. She has gray body color with normal wings, but she's heterozygous. So she's also a carrier for the combination of black body color with these shriveled vestigial wings. We're going to breed her with a male who is homozygous for that second combination. He has black body color and these shriveled up wings. Now, because these traits are linked together, we're not going to see what we would expect here. If we did a Punnett square, we'd expect a 1 to 1 to 1 to 1 ratio. Each phenotype showing that, hey, it's independent of each other. We're going to have equal opportunity for all 4 combinations. We also aren't going to see total linkage with each other, where the individuals who are wild type and the individuals who are black with the vestigial wings, there's only those 2 types. We're not going to see that. What we see instead is that the vast majority match their parents. Either they're wild type, they have gray bodies and regular wings, like the mother, who passed on the dominant alleles, or they're black-bodied with vestigial wings, like the father. Remember, this is technically dihybrid. There's 2 genes in play here. You would expect there to be 4 phenotypes, and there are 4 phenotypes, but the recombined phenotypes are less common. The gray and vestigial, we only have about 200, as opposed to the 950 wild types and black vestigials. Same thing with the normal wings and the black colored body. We see about 200 of them. So we can actually add all of those up together. If we give it the proper numbers, there's just shy of 400. We have 391 recombinants out of a total 2,300 offspring. 391 divided by 2,300 times 100% gives us about a 17% crossover rate. So we know that the likelihood of these 2 genes getting independently assorted from each other is about 17%, which is much lower than the 50% you would expect if they were on 2 completely different chromosomes.